So hello and welcome to MDC Connects 2021. I'm Sarah Brockbank. I'm the lead scientist at Medicines Discovery Catapult, and I'm hosting the session today. I'm with my colleague, Darren Holmes, who's behind the scenes driving the Zoom. So this year, MDC Connects is focusing on complex medicines. And we're joined by a whole host of experts from across the community who are taking us step by step from concept through to the clinic. This week, we're on our fourth webinar in the series. And for those of you that haven't been able to join us for the first three, these are available as recordings on the MDC website. But for today, in this session, we're going to be looking at different delivery technologies and how these are being developed as delivery vehicles for complex medicines. The advantages and the applications of each will be illustrated, and the challenges that need to be overcome to achieve success. We're going to hear the progress being made to develop them into medicines. And I'd like to welcome three speakers. It's an all-female cast this week. So firstly, we've got Yvonne Perry, who's Professor in Drug Delivery at Strathclyde University. And Yvonne's going to talk about lipid nanoparticles to facilitate the delivery of drugs and vaccines. Then we have Louise Coletta, who's the group leader in the Leeds Institute of Medical Research at St James's University Hospital. And Louise is going to tell us about the development of therapeutic microbubbles for targeted drug delivery. And finally, we have Helen McCarthy, who's Chair of Nanomedicines at Queen's University Belfast and also CEO of Phion Therapeutics. And Helen's going to demonstrate how nanomedicines can offer alternatives for the delivery of RNA vaccines. So we will take questions at the end of each of the talks. So if you would like to ask a question, please put this into the Q&A box at the bottom of your screens. Please don't put it into the chat box, please put it in the Q&A box. That's where I will look at the end of each talks. And please type this in while the speakers are talking so that we're ready to take questions when they've finished speaking. So without further ado, over to you, Yvonne.
There we go. Okay, hopefully you can see my screen and hear. Perfect, thanks for that. Yes, so I'll be telling you the work we've been doing on lipid nanoparticles, and the point of the joke is I heard so often described on the news that these are little fat blobs, and you know, as a scientist it does get a little bit stressful when you realise how much work goes into making these little fat blobs, as they're called. So the background for the nucleic acid vaccines, or the mRNA vaccines that some of us have been getting, if you've got the Pfizer or the Moderna vaccine, comes from early work in the 90s. So it was work by John Wolfe, and Felger was the PI on it, where they discovered that if you inject a plasmid DNA intramuscularly, if it encoded an antigen, you actually got an immune response. And that was great, and over time that's been further developed. And rather than injecting the DNA, we can now inject mRNA, and mRNA can encode for a protein antigen, as we see with the COVID antigen, and then we get an immune response based on that. And it's a very useful way of generating an immune response. And the key thing with using RNA versus DNA is when we use DNA, you need to get the plasmid all the way into the nucleus. But if you will go with RNA, you've got one journey less, so you just need to get it into the cytoplasm. But in both cases, we've been looking at using transfection agents or delivery agents. So in this case, I'm talking about the use of lipid nanoparticles to help the delivery of these systems. And why do we need delivery systems? Well, it's very much because RNA is easily degraded. It's highly negatively charged, it's highly hydrophilic, so it's difficult to get into the cell, but it's also degraded by RNA. So you can see here a gel electrophoresis, some RNA before it's digested, and then we mix it with an enzyme and it's degraded very quickly. And indeed, you actually need to watch when you're handling it in the lab, so you need to wear gloves because we have RNAs in our hands and in surfaces, and it will actually degrade if we don't use appropriate protection against that. So you can see it's an easily broken down molecule, so it does need delivery, but we know it works. We're all seeing the benefit of that in the current vaccine programme that's being rolled out. So that's good. We have this system. So there's various types of nanomedicines. Liposomes have been around since the late 80s. In terms of approved products, we do have liposomes, and the difference being we have this lipid bilayer made here, but with liposomes we have an aqueous core, and the drug can go into a core or into the lipid membrane, shown here. And this is a cryo-TEM of images of liposomes we've made, and you can see the bilayer with the aqueous core. In contrast, lipid nanoparticles quite often have very similar uses and similar lipids being used, but we do use an ionizable lipid to bind to the RNA, and this is a schematic shown here. There is some debate over the overall structure, but the schematic helps you visualize it. So you can see the RNA in here, it's bound to the lipids, and the whole thing condenses down into a smaller particle. And you can see the cryo-EM, it's much more zoomed in. I appreciate they look bigger, but much more, they're about 50 to 100 nanometers in size with no notable aqueous core because it has complexed and shrunk down around the RNA. So there is some differences between a liposome and an LNP, basically structurally. And what do we need from an LNP for it to be effective? Well, we need various things. We do need protection. It needs to protect the RNA and not allow it to be broken down. We do need for it to be potent, so it needs to be able to deliver the mRNA and release it and get the transfection. It needs to also control biodistribution or cell uptake. Where we are in that control side depends what you want it to achieve. And also the other thing is we do need to think about manufacturability. So people always think, we'll think about that last, but if you can build it into your process early on, it makes life so much easier. So it is a key thing to be thinking about what is the plan for this manufacture? How are we going to make it? And making sure we can actually scale up is an important aspect. So in terms of lipids, so it really came from the starting block, most people would say, is cationic lipid DOTAP or similar. There was cationic lipids used, and they were being used for the DNA vaccines. However, you've got an overall cationic charge, and as we know, if we go too cationic, we can actually cause cell lysis because you have an electrostatic interaction. So there was a move to the more neutral lipids, and we see two examples here. So the variation, so you can see they have an apparent pKa about 6.5, 6.7. So if you take them 2 units below, then they're going to be fully ionized. If you take them above, then they'll be neutral. So the idea is you work with them while in the cationic state at lower pH, they can bind your RNA, and then before you make it into the human, you'll change the buffer, so you change it into PBS, brings up your pH, so it makes the overall LNP near neutral. So you get over that problem of injecting something cationic. So the combination of LNPs is basically shown here. They're all roughly the same. So they tend to have about 10% distereophosphatidylcholine. That's a structural lipid and it helps it stabilize the particle. Cholesterol, there's around about 50% in the formulation. Again, that holds the structure. Then we have an ionizable or cationic lipid to bind the RNA. And then we have a pegylated lipid to help stabilize the system. And depending on the various vaccines, the ionizable lipid and the PEG lipid really are part of the IP package of these vaccines. So those are the two that vary, but at the moment, the majority of them are using DSPC and cholesterol. So in terms of production, what you tend to do, there's variations in a theme, but overall, generally, you've got your lipid in a solvent. So the lipids we talk dissolved in mainly ethanol. You've got your RNA in a buffer, aqueous buffer, normally at a slightly lower pH. You mix them together, you basically increase your solvent polarity, so you cause the lipids to come out of the ethanol and they form particles because they're not water soluble. So they form these particles together and then we can bring up our pH. So in our lab, we've been working a lot with microfluidics to achieve this. And two of the microfluidic cartridges, which are from Precision Nanosystems, we work a lot with that team. And you can see here, this is a staggered herringbone. So it's basically good grooves and it basically folds the solvent and the aqueous phase together. And as that happens, you get nanoprecipitation and the particles form. We've also looked at a more recent development that P&I made as the toroidal mixer, which is shown here, and it basically mixes it a slightly different way, but they've developed this one because it's easier for them to scale up, and we've been using it in higher manufacturing. In microfluidics, there's basically two key components. It's how we mix, the ratio mix of the aqueous to the ethanol, and how fast you run the system. Those are the two key things. And the parameters that are important for nanomedicines, I'm sure you're all familiar, your size and their distribution. You don't want very heterogeneous mixture. Zeta potential is important for the LNPs that we've been looking at. You want to make sure your lipid comes out and doesn't get stuck in the cartridge and lipid fouling. You want to make sure your mRNAs are loading and it can come out at some point, and it should, as I say, have potency. So we did some studies on these systems, and we were basically looking at the original ionizable lipid, which is D-lin MC3-DMA, as used in a commercial product. And we also compared it to two easily available cationic lipids. So these were easy for us to buy in the lab and much cheaper than the D-lin. So it's a great way of getting going with LNPs, getting a feel for them and checking how they work. And then once you've optimized your parameters, you can substitute in the much more expensive lipid. And we used it in the usual combination with distereophosphatidylcholine, cholesterol, and the pegylated lipid. The first thing was to check our manufacturing processes. So, as I said earlier, how we mix them together, aqueous and lipid, and you can do them at a 1-to-1 ratio. You can do it at 3 aqueous to 1 lipid, or 5. I mean, you can do it any ratio you want, but those were the 3 we tested. We had it with our cationic lipid, MC3, which is the ionizable, and then DDA, which is the other always cationic lipid. And we measure particle size, and the circles are here are PDI. So you want a PDI below 0.2, as you see with the dotted line, that's kind of threshold. And what you can see when we mix it a 1-to-1 ratio, that's, you know, we're not getting good particles, but anything above 3, so 3 aqueous to 1 lipid phase, we get nice small particles, low PDI. And then as we go higher, there was no further advantage. In terms of how fast we could run the system, so we were using the benchtop nano assembler, and it runs between 5 and 20. And with the LNPs, you do see a slight drop in size as we increase the speed. Anywhere between 10 and 20, we got reproducible results. And you'll see we have scaled that up even faster. So we knew we could make them. We knew how to control the size. We obviously had to check we could get loading. So we tested poly A, again, as a surrogate for mRNA, much cheaper, single-stranded DNA, and mRNA, more expensive. And basically for all three, the poly A, the DNA and mRNA, good loading irrespective of using a cationic lipid or the ionizable lipid. So everything goes in, no big surprise, you would hope it did based on the system. So we checked this scaled up. So these are empty LNPs, so they're a bit smaller because they have no mRNA in them. Then we used our standard benchtop nano assembler. We then swapped to the new toroidal mixer that allows us to scale up, so that's here. And basically you're looking at size on the left, PDI on the right. And once we put the mRNA in it, the size is reproducible, PDI is nice and low. We then went to a faster speed, so we're running here at 12 mils per minute. Now we're up to 60 mils per minute, reproducible, so we can go from bench to GMP production, which is up at 200 mils. So basically we took our process parameters that we developed in the lab, and it goes straight onto the GMP system with it all mapped across. In terms of loading efficiency, again, completely mapped across. So that was great. We could see from bench to basically bedside, to use that acronym, we were getting a nice reproducible process. And the other aspect to this, and I know that CPI are going to be presenting later, they went on and have developed this further to build in inline processing, which includes removal of the solvent. So they've done a lot of really nice work on the removal of the solvent, which is part of the process. So we wanted to then check the potency. So we took our 3 LNPs. So one's made with the cationic lipid DOTAP, one's made with the cationic lipid DDA, and then the ionizable lipid MC3. We took them in vivo, so with mice, groups of 5 were either injected with 1.5 or 0.15 micrograms of self-amplifying RNA, which is a slight variation in the mRNA, and you can normally use lower doses. And it encoded for a rabies virus glycoprotein G, so basically a rabies virus system. And we used that because there is a good benchmark, and also we were working on this prior to COVID. So the mice were injected twice, 0 and 28, and at the end of the study measured the usual antibodies, cytokines. And I'll mainly focus on the antibodies today because you get the similar story running throughout. So here we have our antibody responses. So the good thing about, as I said, we used the rabies vaccine protocol because there is actually a commercial product, Rabipur, available. So it's not an mRNA, but it's a good benchmark. So it's one that's used in humans, and for mice you use 5% of HD human dose. And then the lower line is the limited detection for the assay we used. The upper line, because we have human data that we can relate to, this upper dotted mark means protection. So any results you see above that means you would get protection from this vaccine. So this is 2 weeks after the first injection. So you can see the commercial product working well in the mice. And then you've got our DOTAP and DDA cationic lipids and the commercial ionizable lipid. And you see good responses across them all, no significant difference, except at the lower dose with the ionizable lipids. So that's great to see. With much cheaper lipids, we can get a very similar response. We then moved to 2 weeks after the second injection, all the immune responses have gone up roughly 20-fold. But you can again see a very similar profile for all of them, and really it's only at the lower doses we can tease out differences. So the ionizable lipid is working better at the very low doses, but we certainly see good responses from the easy accessible cationic lipids. So if you're looking to develop mRNA encoded vaccines, the easy to use cationic lipid platform is available and you can buy that. But there's also obviously lots of different ionizable lipids available. So next thing I wanted to check was biodistribution. Do these act differently, the cationic versus the neutral? So we basically fluorescently labeled our nanoparticles, injected them into the mice and followed them, the biodistribution. So you can see lots of pictures, but basically you get a cationic ionizable, a cationic lipid DOTAP, cationic DDA, and the ionizable LNPs. You get time running down from just after injection up to 10 days. And the fluorescent that we've circled here is basically where the LNPs are sitting. So we inject them IM, and you can basically see it sits at the injection site, very similar for all of them. There's not a major difference. And if we plot it as total flux, we can see that the ionizable lipid and the DOTAP, very similar profile. The DDA sits slightly longer. And if you calculate its area under the curve, you see a similar thing. So one of the cationic lipids sits at the injection site slightly longer than the other, but that certainly didn't translate back to better immune responses. So we are seeing here that biodistribution and vaccine efficacy are not directly correlating at this stage. So that was a good thing to learn as well and to look at. So in summary, what we could see is we can manufacture LNPs. We've got a route from bench straight up. So the idea is by scaling early and checking that, you can enhance your speed through that, but also you do have to check your potency. We did also do cell work, but really there was very little indication of correlation. So that is a gap in the market we still need to address better, and well, we in our lab need better in vitro models in that respect. And just to say, this work was funded by the European Union and in collaboration with GSK, it's passed stream back. And Despo, Julia, Gustavo and Rob were instrumental in many of the in vivo studies. And Carla, who was our microson, Carla Rossi, works with CPI, or we worked with CPI in developing the Scaleup model. And thank you for your time.
Take the mute. Thank you, Yvonne. So some questions in for you. So how much benefit, so actually this is a question that I probably pose to all of the speakers, but how much benefit could an optimized lipid mixture make to current and future vaccine manufacture?
That's a good question. So obviously the vaccines we've got are working very well. We don't know if we can optimize it more. I mean, there's always route to optimize and improve things, and it may not be LNPs are the only formulation. So we did some work where we were looking at different routes of administration, and when we tested these formulations we were looking at intranasally, they don't really work well. So we're definitely learning lessons that, I mean, it's kind of always on hindsight, but you do have to optimize your formulation for your given route. So these were only the data only shows IM, but if they didn't work well intranasally. So yes, we'll probably have to use different formulations. It may not just be LNPs that are the best way forward. So I think there's still a lot to be learned in this area, and I'm sure we can improve further on where we are with them.
And how versatile are the nanoparticles for you to carry out drug design and optimize for different therapeutics, different indications?
Yeah, so the good thing, well, this is what we're hoping at the moment, is with the mRNA, it's pretty much plug and play. So we hope we can just snip out the encoding for one antigen or protein or whatever you need, put in another one. If you've got your manufacturing process set up and you keep your L/N/P ratio, basically your cationic to anionic ratio the same, and it works, then you can just swap out one, swap in one. And that's the hope, and I know that's been some of the discussion about if we need to change variant. It should be that the drug substances, we call it the mRNA, if it's got a lot of the same process parameters, and then it should swap in and out easily. So that's the hope from LNPs, definitely. And I think we are challenging that question now going forward. I think it's really useful.
Okay, brilliant, Yvonne. So over to Louise now to hear about some microbubbles.
Thank you, Sarah. Okay, and thanks to Darren as well, who's due to technical malfunction my end, is going to work the slides, I hope. So in the next 15 minutes or so, I'd like to share with you some of the work we've been doing in Leeds on developing therapeutic microbubbles for targeted triggered drug delivery of cytotoxic drugs for the treatment of colorectal cancer and liver metastases. Next slide, please, Darren. So the impetus for doing this type of research comes from the fact that despite all the improvements in cancer drug discovery and delivery, most cancer patients receive chemotherapy at some stage during their treatment. And this generally takes a one-size-fits-all approach where highly cytotoxic drugs are infused and often with consequent side effects due to off-site toxicity, which can prevent the use of stop the treatment. So in order to overcome this, we and a lot of other people, as you've just heard from Yvonne, have developed nanoparticles to try and improve the pharmacokinetics of drugs delivered in this way. And we heard earlier in the program about liposomes as well. So could I have the next slide, please? And if you click on the left one, thanks, Darren. So we have taken the approach of trying to improve on the pharmacokinetics from those nanoformulations by developing targeted triggered drug development using the microbubble as the basic building block. Microbubbles, as you could see on the video there, I hope, are contrast agents for clinical ultrasound imaging. They are gas-filled lipid spheres of about 2 microns in size, which because of their size, stay within the vasculature. So they are intravascular traces. So we and others have taken the leap to use the microbubble and see if we could deliver drugs with them. And in our instance, the therapeutic microbubble is shown schematically on the right-hand side here. And we're going to play the video. It's got, we attach liposomes to the microbubble shell, as you saw there, inject them intravenously. They flow along the blood system, and due to the targeting molecule on the surface of the microbubble, they bind the tumor tissue. Where the ultrasound destruction pulse is delivered, it bursts the bubbles, and hence we have the targeted ultrasound triggered drug delivery, which hopefully will give a better therapeutic index with less off-site toxicity. Can I have the next slide, please? So in Leeds, about 10 years since, we were lucky enough to set up a group looking at engineering therapeutic microbubbles for cancer drug delivery. Excuse me. And these are shown here. The groups we had in physics were looking at fabrication, characterization, and drug loading. And we had a group in electrical engineering as shown here, who were interested in ultrasound and the response of microbubbles to ultrasound and the effects on drug release and uptake. And our group were involved in looking at how microbubbles could release the drug and improve PK and PD release. So we were lucky to be funded by EPSRC through their healthcare technology platform. And in the final iteration of this, we also were fortunate to have a collaboration with the MDC itself, which brought on board expertise in getting to regulation for first-in-man clinical trials, and also advanced PET imaging, which I'll mention briefly. So the next slide, please. So the group worked iteratively, as I said, in developing and optimizing the basic building blocks for microbubble drug delivery. And as shown here, the microbubble has the gas core with a lipid shell, to which we can decorate it with anything of choice, really. We have a PEG layer, which improves biocompatibility, and a targeting ligand, which is added on the outside of the shell. Next slide, please. The payload which we have developed, which are used to decorate the microbubble shell. For hydrophilic drugs, we have liposomes, which have been mentioned, and we encapsulated 3 different payloads here, which will mention luciferin, which is a reporter for bioluminescence. And the drug we're interested in was irinotecan, is a topoisomerase inhibitor used in treating colorectal cancer. And it is metabolized in vivo to SN38, as shown here, which is 1,000 times more toxic than irinotecan itself. But it is hydrophobic and currently can't be delivered by systemic routes. It's hydrophobic, and in the liposome here, it integrates into the lipid shell. On the right-hand side, you can see the schematic for a drug delivery vehicle called a LOND, which Steve Evans and the physicist developed for a lipid oil nano droplet. And this has an actual oil within the lipid shell in which drugs can be dissolved. And we used combrostatin A4, which is a vascular disrupting agent. But I won't go into the data for that today due to time, so we'll go with the payloads shown on the left-hand side. So in the next slide, please, it just shows you how the two come together, the therapeutic microbubble, the drug loaded onto the shell via streptavidin, neutravudin, biotin linkages. Next slide, please. So having designed what we hope was a vehicle for drug delivery, we had to be able to manufacture these for our experiments around the 3 groups in Leeds, but also with a view to scale up for future clinical trials. And Yvonne just introduced perfectly microfluidic technology, and we use the same approach here. So you can see in the schematics, the microfluidic chips, which we used, and we flowed gas and the lipids within them. So the bubbles self-assembly, as you can see in the right-hand side, to form the microbubbles, which give this milky appearance in the tube. Now, the innovation in Leeds in the bottom video, which is playing, was to develop the chips so that they had this milled region. So microbubbles were produced in a microspray regime, and that's what's shown here in the video. And using this regime, it produces a relatively monodispersed population, which we've heard is good for clinical use and drug delivery. If you click, please, Darren, you can see here how we've developed multiplexing of the chips with an eye on producing large numbers for experiments and for translation to the clinic. Next slide, please. So that was the basic and innovative technology used to develop the microbubbles themselves. So the next step was to produce the therapeutic microbubbles on chip as well as a way to producing them. So that is add in the payload, as there's the liposomes and the targeting antibody. And in the schematic below, you can see how these are flowed through the chip. Click, please, Darren, to produce therapeutic microbubbles with concentrations in 10 to the 9 bubbles per mil, which are antibody targeted, and as I said, have encapsulated irinotecan or SN38. Next slide, please. So when we produced these different iterations of the microbubbles, we tested them in each of the groups, either for their physical properties, for their ultrasound characteristics, and we were looking at their in vitro and in vivo assessment in terms of various parameters. So the first assays we developed here were for looking at targeting of the microbubbles to the tumor vasculature. As the microbubbles are intravascular tracers, the target to which they're going to bind in the tumor vasculature has to be on the endothelial cells, and VEGF receptor 2, vascular endothelial growth factor receptor 2, is upregulated in human colorectal cancer and in the mouse xenografts. And as you shown here in the schematic, the top right is an Antibody chip in which we can flow the microbubbles and look at their binding to endothelial cells, as shown below, giving us a more realistic idea under flow conditions of how the microbubbles will behave. Next slide, please. So having shown that the VEGF receptor 2 targeting could facilitate binding to the endothelial cells, we looked at whether this could also mediate and increase drug release from reported payloads. So just in this slide, it shows the data from an experiment where we used luciferin encapsulated in liposomes and attached to microbubbles. As the luciferin is very leaky, we didn't, there was no other ultrasound trigger at this time. We delivered the microbubbles to the mice bearing human colorectal cancer xenografts expressing luciferase. So where they receive luciferin, you get the bioluminescent signal seen. And in the quantification on the right-hand side, you can see that where we had VEGF receptor 2 targeting, there was increased release of luciferin detected in the tumors. Next slide, please. So, having shown that our targeting could effectively work, we moved on to the actual chemotherapeutic drug, first one being irinotecan. And we loaded the drug-loaded liposomes onto the microbubble shell and used the schematic for an in vivo experiment shown in the diagram at the bottom of the slide with the 4 groups highlighted in the colored boxes to look at the effect of incorporating an ultrasound trigger into the targeted delivery with the therapeutic microbubble. Click, please. And when we did this and analyzed the response in vivo, which was in terms of tumor size measured accurately using high frequency ultrasound, we could see that in the group that received the therapeutic microbubbles with the trigger, the ultrasound trigger, there was significantly reduced tumor volume. So this gave us the confidence that this type of delivery system using low dose chemotherapy drug with an ultrasound trigger could in fact have an effect on tumor growth. Next slide, please. How this might be working, we looked at looking at the biodistribution and the metabolism of the therapeutic microbubbles in vivo. So the same groups, we'd collected tissue after 5 treatments, 3 days post the final treatment, harvested the tissues and developed a sensitive liquid chromatography tandem mass spec technique to detect the drug and its metabolites in the tumor itself. And interestingly, what we found, if you look in the bottom right-hand graph, was that after 72 hours post-final treatment, the drug irinotecan and its metabolite was only detected in the group which had received the therapeutic microbubbles with the ultrasound trigger. There was in any of the other groups, it was any drug was below the level of detection. And in that same group of microbubbles plus trigger, there was also irinotecan and SN38 could be detected in other tissues of the group as well. Now, one of the things about using encapsulated drugs and analyzing them is that the tandem mass spec can't distinguish between the encapsulated and the unencapsulated free drug. And so the last bar chart on the bottom right-hand side shows the relative amounts of irinotecan and SN38 in tumor and tissues to try and get a handle on how much of the drug is actually being released and is free and available to bind DNA in the tumor cells. And you can see that it's mostly in the tumor and also in the colon, where we know the drug is being eliminated, and that's the free drug. Next slide, please. So we know that SN38, as I said, is far more potent than irinotecan itself. And as I think we showed in the previous experiments, therapeutic microbubbles can deliver drug at low concentrations. So we decided to encapsulate SN38 itself and use it in targeted triggered drug delivery. So the schedule that we used is shown on the right-hand side, and as shown in the graph below, you can see that we got significant inhibition of tumor growth using this drug in this drug delivery platform. And with the aid of Juliana Maynard at the Catapult, who performed some radio tracing PET experiments, which she's going to talk about next week. If you click, please, Darren, I think the conclusion from most of this work that I've rapidly tried to show is that therapeutic microbubbles can improve the therapeutic index of cytotoxic drugs by increasing the percentage injected dose reaching the tumor, but also importantly by limiting the bioavailability in normal tissues as well. Next slide, please. So just to wind up, bursting with promise, our next stage is to translate therapeutic microbubbles to the clinic, and with the aid of the Catapult, we have moved to gain regulation to do this. The final slide is just the names of the wonderful people in our microbubble group who have done all of this work, of course, in Leeds and Bradford. And thank you.
Lovely, thanks, Louise. Just time for a quick question on this one. One question here, is there a risk of air embolism from the microbubbles?
No, I don't think so. They're injected, the gas in the microbubble is perfluorocarbon gas. Sorry, I probably didn't say that, but it's a perfluorocarbon gas which is soluble, it dissolves, and it is breathed out through the lungs.
Okay, brilliant. Thank you very much, Louise. So now over to Helen McCarthy from Phion Therapeutics.
Thanks very much, Sarah, and good afternoon, everybody. Thanks for giving me the opportunity to talk to you today about the technology that we're developing in Phion Therapeutics. It fits very nicely after the first two talks that we have been lucky enough to hear this afternoon. This is about alternative delivery for messenger RNA vaccines. So a little bit of background as to how Phion Therapeutics came about. Okay, and let's get the slides going. Okay, so Phion have developed a peptide called RALA. Now, this originally came out of my research group in the School of Pharmacy in Queen's University, Belfast. So as a young academic, you always get asked the question, you know, what are you going to start your research group on? And back in the day, I was very much into delivery and very much focused on non-viral delivery. And so there's been a lot of research that has underpinned this, many research articles, and as academics do, you search for the funding and you get as many grants as you can to take the technology as far as you possibly can. For me, there came a point in 2017 where if we really wanted to try to make this into the clinic, we felt that we had a strong enough position and enough data to spin out, and that's what we did. So the birth of Phion Therapeutics happened, and we are a preclinical stage therapeutic vaccine development company, where we are developing the most promising preclinical assets. We have 3 main leads at the moment. Now, note that it's positioned as a therapeutic vaccine, and I'm going to come to this quite a lot throughout the course of what is now left as my 13.5 minutes in this talk. And that is because you want a very different immune response for a therapeutic vaccine than what you would want in some of the talks that Yvonne gave and in the COVID vaccine, where it is very much about sort of getting that antibody response as well as the T-cell response. For a therapeutic vaccine, we really exclusively want a therapeutic CD8 T-cell response. So we've been very fortunate. We've been sponsored by Innovate UK, have been great supporters of the technology, and that money has really helped us develop our 3 lead programs. We also work closely with Medicines Discovery Catapult. In fact, Sarah was the scientific advisor on some of the data that actually I can't show you today because it's so good that my IP lawyer has said, no, you're not allowed to show that data, but don't worry, you'll get the flavor. And so we really have worked with experts, and I think that's really important. Coming into the Alderley Park, even as sort of, you know, a virtual seat, has really helped us tap into the expertise there to get this to where it needs to go. Okay, so what's it all about? I gave you the sequence in the first slide. It's a 30 amino acid peptide. It is designed to overcome the biological barriers. It is positively charged. Some of you will have noted that it has arginine on the backbone, spaced out in such a way that it is bathed with a hydrophobic leucine. It can condense anionic cargo, nothing new there, but the way that it condenses it down is it intercalates with it like this. So, and really there's cysteine residues at the end of the nanoparticle that will form the cysteine bonds and make the nanoparticle really tight. We get really good size and charge characteristics to make it ideal to enter cells. Again, for those of you in the drug delivery spectrum, you may go, okay, fair enough. But where this is really different is that it enters the cell without the cell even realizing that it has entered it. And what I mean by that is we've done work with cell and gene therapy catapults, and they have shown really nicely that when we enter the cell, we don't change the receptor status in the cell in any shape or fashion. And that's really important because we almost enter in a stealth-like mode. Now, typically our particles will enter via clathrin-mediated endocytosis. Obviously then, once inside the endosome, the pH drops. When the pH drops, it actually makes the peptide change from more random coil to more alpha helical. Hence, pH responsive delivers ions for Phion. And what that does is it fuses with the endosomal membrane and allows a highly efficient escape of the cargo. Again, doing it in a stealth-like fashion where it's not evoking any toxicity. So why is this applicable, particularly for therapeutic vaccines? Well, this is our pipeline. We started off with the mucosal HPV vaccine. And the reason that we chose this was because the E6 and the E7 antigens are really well known. And we wanted to show that we could get into a really high percentage of CD8 T cells and that the response would be, really at a competitive level to others that have done this. Note that most of the other HPV liposomal vaccines have to be delivered intravenously. And Yvonne talked about this at the start. Some vaccines will be delivered intramuscularly. Some that are trying to be therapeutic will be delivered intravenously. These ones are delivered intradermally. So these peptide messenger RNA nanoparticles cannot be delivered intramuscularly because that's not what they're designed to do. They're specifically designed to enter the antigen presenting cells that reside in the dermal layers of the skin. As I said, we want a CD8 therapeutic response, very important. We do exclusively get that CD8 response whenever we use RALA to deliver the messenger RNA. And it's achieved by the stealth mode delivery to the antigen presenting cells and in a way that does not provoke a cellular innate immune response. That's important because we don't want our nanoparticles to be cleared. The other thing as well is we're not restricted on the amount of cargo that we can condense down, so we can deliver one antigen, multiple antigens. And what's really interesting with this is that the way that the peptide condenses down the nucleic acids, it really protects it. Believe it or not, that following post-lyophilization, our vaccines do not require cold chain storage, so we can store our messenger RNA vaccines on the bench. This was a landmark study, and the reason why I'm really flagging this up is I think it exemplifies why this is a little bit different. On the left-hand side here, this was work that we did with Ghent University and published in Advanced Healthcare Materials in 2017. These mice are genetically engineered to show the luciferase production under the control of the interferon beta promoter. So whenever we inject our nanoparticles, if we get luciferase, there's production of interferon beta. We show really nicely that whenever we give our RALA messenger RNA nanoparticles, that there actually isn't any production of luciferase. Therefore, there's no activation of interferon beta. And we've done numerous other studies to show this. And you can compare and contrast this with the uncomplex messenger RNA and again with the cationic DOTAP messenger RNA. What this means is that the messenger RNA, the cargo, does what it says on the tin. There's nothing in the delivery system that's flagging this up as something foreign that's going in. Now, this was delivered intradermally. And what we did then was we translated this into interferon knockout mice and wild type mice. So what we wanted to show was the percent specific killing. You have to make sure that you do get a really good high percent specific killing in order to clear an infection, or in the case of HPV, to try to regress some of the lesions. And you can see very clearly that there's no significant difference between those mice that are wild type and those mice that have interferon knockout, i.e., basically the immune response was the same in terms of the CD8% specific killing, irrespective of whether a full immune response was there in the wild type mice or whether we had knocked out the interferon. If you compare and contrast this, you can see that there is a difference here whenever we look at the DOTAP with modified messenger RNA. Only whenever the interferon is knocked out do we get comparable levels of percent specific killing. So it exemplifies really nicely how peptide delivery of this messenger RNA works really nice for intradermal delivery. What we then went on to do as start of our preclinical program for this for HPV, this was a biomedical catalyst grant sponsored by Innovate UK. We labeled the messenger RNA with Cy5, and we looked to see where they go once we give them the site of administration. So in the mouse, we inject into the ear of the mice intradermally, and we were able to track where the nanoparticles go in the draining lymph nodes. There is a high proportion of dendritic cells in the intradermal layer, and some of those are migratory, the CD11C plus ones. And what we show really nicely is at 2 hours there, you can see that there is presence of the Cy5 in the lymph nodes with the RALA Cy5 messenger RNA versus the naked Cy5. You absolutely need to have your dendritic cells migrating to your lymph nodes if you want to start talking to your T cells. And that's what we want in this therapeutic application of a vaccine. And when we took the lymph nodes out and we had a look at this, again, you can see quite nicely that we do get a really nice high percentage. Well, trust me, that more than around a couple of percent of CD11C positive cells for your cargo is high. And you can see that you get a good expression, and then it goes down again at 96 hours. So we're getting to those draining lymph nodes, and we are certainly getting into the antigen presenting cells that we want. We have gone on then to look at the immune response. So we have done a prime boost in mice, and we've pulled out the splenocytes at the end and then stimulated with the E7 peptide to make sure that we do get a strong CD8 response. It is exclusively a CD8 response. It is interferon gamma positive, and that has translated very nicely now into our therapeutic tumor models. We've gone for HPV because it is a big problem. We know that there are vaccines out there, Gardasil, Cervarix, that are very good at preventing young people from getting HPV, but for around the 200 million people worldwide that currently have HPV, the therapeutic options are somewhat limited. And really 90% of the human papillomavirus is caused by HPV 16 and 18. So those really are the ones that we're going after with this. We are underway with one of our later preclinical studies before we go into toxicology, where we have given the mice these tumours, and then we've done a prime boost. And we're very happy to say that the CD8 response has translated into regression of those tumours. This is our plan, and again, I reference Alderley Park, and I certainly reference Aptus Clinical, who are helping us very much with this. Having not done this before from the School of Pharmacy in Belfast, it's very important to tap into that expertise and realise what your limitations are. So in respect to that, we're going to do a module 1, module 2 type of clinical trial that we're planning out, and CETA are actually helping us with the pharmaceutical development of this, and Apconics are going to help us with the toxicology. I said earlier, we have lyophilized this into a product with sugars, and you can blend your sugars to improve antigen presenting cell uptake. We've stored this at room temperature, and we also get functionality again at 6 months post lyophilization, and we're taking this out further. Yvonne mentioned, again, the Nano assembler. We have been fortunate enough to get a continuity grant together with the Centre for Process Innovation, where we started to look again at the early stages of how do we manufacture this up. And just to emphasize some of the points again that Yvonne said, sorry, I sound like a broken record, but we're all singing from the same hymn page on this. Manufacture is critical. It's critical at the start phase where you add A to B, and how are you going to then scale this up? And what we are doing here is adding the peptide to the messenger RNA in water. Is a very green technology. And we've used the benchtop nano assembler basically to optimize the flow rate and the parameters required for microfluidics. So you can see here the difference. The legacy method, as we quote here, and we did this with an exemplary siRNA cargo for this project. You can see the difference in the spectrum between the legacy method versus the automated production on the Ignite system. Again, in much nicer peaks and better distribution in this. And what was nice was once we optimized the parameters in-house, we then sent those parameters over to CPI to see if they could replicate this. Tech transfer is critically important in Scaleup, and very pleased to say that they were able to get 95% plus encapsulation efficiency of the cargo. The reason why there's an extra up to 5% is because of how it condenses down the cargo like this. So you will always have little bits of messenger RNA that can be sometimes detected as unencapsulated. What was also very exciting for us is that in 3 different test runs, and somebody that works in electrostatic nanoparticles, I got very excited by this because each of the test runs you can see here, we were able to get a Z average that was very incredibly similar from 58, 64, 63, and PDI levels at this stage, which for us is very good at less than 0.3. So just to summarize, before Sarah kicks me off this talk, we think this is very versatile. It isn't for all types of vaccines, but it is very good for therapeutic vaccines, and it's very efficient and safe, and it's also incredibly stable. I didn't do any of this on my own. I'm working with a large team, Dara, John, our chairman of our board, and a lot of people helping us to get to where we need to go. Thank you very much for your time.
Thanks, Helen. Superb timekeeping today from everybody. Lovely. So questions on this one today. At what stage did you start thinking about manufacturing?
I actually started thinking about it very early on whenever I was an academic. Prior to working in the peptides, I was working in recombinant proteins that you would make, and you would have to make those, you know, through expression systems, and then you would have to purify through columns, and then you would have to desalt. And I was thinking, this is not the way to go to try to scale this up. So I think you have to think about it whenever you're developing the technology at a very early stage. I think I could be slightly biased. I think I'm sort of fortunate. I think Yvonne's also fortunate. When you work in a school of pharmacy, you're thinking of the final product right at the very start. And I think that helps a lot. But one of the biggest, I think, advantages of this technology is how easy it is to make them.
I've got another question here, which is from your pipeline, you were working with small molecules as well as with nucleic acids. Is the RALA the same?
It is the same. Yes, yeah, absolutely. It's all related to the charge of whatever cargo that you want to deliver.
And very similar question then, for the different therapeutic areas that you're working in, that would suggest that you're able to target different cell types.
This is something, again, that I think you have to be realistic about. Depending on the indication, depending on the cargo, depending on the route of administration, no one delivery system is going to solve it all. You know, the complexities of each delivery system will have a sweet spot. We've identified a sweet spot for CD8 vaccines. We've also got a sweet spot we can deliver to some quite nice cells in the lung. And again, that's why, you know, we potentially are looking at some other cargo there as well. I think there will be a suite of ideal nanomedicines for different indications, and it really isn't one size fits all. You have to be really pragmatic and realistic about that.
I think that's a perfect answer to finish the 3 talks that we've had today, actually, Helen, isn't it? That shows why we need the different technologies. So thank you very much for that. I will share my screen. So thanks for all 3 speakers today for 3 really excellent talks. Next week, we're moving on now to understanding safety and efficacy, and we have 3 speakers, Juliana Maynard, who's been mentioned today in the talks, and Juliana's going to be talking about the physicochemical characterization, new and novel approaches to understand the PK of complex drugs. And we're joined again by Jenny Worthington from Axis Bio, who is going to be talking about determining efficacy in vivo and specifically addressing the challenges of working with complex medicines to do this. And finally, Richard Knight from Apconics is going to answer the question, do complex medicines raise different challenges from a safety perspective? So I hope that you will join us again next week on Tuesday at 2 o'clock again. And thank you once again for our 3 speakers today. Thank you. Goodbye.